Hello everyone, welcome to Explore Biology. Today we will discuss the topic bacteriophage. The word bacteriophage is derived from the Greek which means to eat bacteria. Bacteriophage are also called bacteria eater. The viruses that infect or eat bacteria are called bacteriophages. Bacteriophages can be categorized into two types, virulent and temperate. Virulent phage enter the lytic pathway that uses the metabolic machinery of the host cell to produce progeny viruses and kills the host. Example is Bacteriophage T4. Temperate phage either enter lytic pathway or lysogenic pathway inside the host cell. If it enters lytic pathway, then it kills the host cell like Bacteriophage T4. But if it enters lysogenic pathway where its genome gets replicated along with the host cell genome during each cell duplication. Example is Bacteriophage Lambda. Both of these viruses infect E. coli, so called as coliphage. Let's talk about the structure of Bacteriophage T4. Bacteriophage T4 is a large virus having a double-stranded DNA molecule packed inside a proteinaceous head. The T4 chromosome is approximately 1,68,800 base pairs long. The tail of the virus contains several important components. Its central hollow core provides the channel through which the phage DNA is injected into the bacterium. The tail sheath functions as a small muscle that contract and push the tail core through the bacterial cell wall. The 6 tail fibers are used to locate receptors on the host cell, and the tail pins on the base plate attach firmly to these receptors. All of these components must function correctly for the phage to infect an E. coli cell successfully. This is the structure of Bacteriophage T4, which has a double-stranded DNA molecule inside the proteinaceous head, hollow tail core through which the phage DNA is injected into the host cell, contractile tail sheath that contract and push the tail core through the host cell wall. 6 tail fibers are there that are used to locate receptors on the host cell, and the tail pins on the base plate help to attach to the receptors that are present on the host cell. Life cycle of Bacteriophage T4. Bacteriophage T4 is a lytic phage. When it infects a bacterium, it replicates and kills the host, producing about 300 progeny viruses per infected cell. After the phage DNA is injected into the host bacterium, then within 2 minutes, it starts synthesizing the proteins that shut off the transcription, translation, and replication of bacterial genes, and the virus take control over the metabolic machinery of the host. Some of the phage genes encode nucleases that degrade the host DNA. Other phage proteins initiate the replication of phage DNA. At about 17 minutes after infection, the assembly of progeny phage begins, and the infectious progeny phage start to accumulate in the host cell. At about 25 minutes after infection, a phage-encoded enzyme called lysozyme degrades the bacterial cell wall and rupture the host bacterium, releasing about 300 progeny phage per infected cell. T4 DNA contains an unusual base 5-hydroxymethyl cytosine HMC instead of cytosine. Derivatives of glucose molecules are also attached to the HMC. These modifications protect T4 DNA from degradation by the nucleases that it uses to degrade the DNA of the host cell. This figure shows the life cycle of Bacteriophage T4, that is lytic pathway. In step 1, a T4 bacteriophage attached to an E. coli cell and inject its DNA. In step 2, 2 minutes after infection, synthesis of phage-specific mRNA begins. In step 3, 6 minutes after infection, replication of phage DNA begins and host DNA has been degraded by phage-encoded nucleases. In step 4, 14 minutes after infection, DNA-filled heads, tails lacking tail fibers, and assembled tail fibers first appear. In step 5, 17 minutes after infection, the first intact phage particles are assembled. And in step 6, at about 25 minutes after infection, the host bacterium is lysed, releasing about 300 progeny phage. These progeny phage infect another bacteria, and this cycle continues. Bacteriophage lambda. Bacteriophage lambda is smaller than Bacteriophage T4, but its life cycle is more complex. The lambda genome contains about 50 genes in a double-stranded DNA molecule, which is 48,502 base pairs long. This linear DNA molecule is packed in the head. Soon after it is injected into an E. coli cell, the DNA molecule is converted into a circular form, which participate in all subsequent intracellular events. The structure of bacteriophage lambda consists of head and tail. Double-stranded DNA molecule is packed inside the head. Life cycle of bacteriophage lambda. Bacteriophage lambda can enter either a lytic pathway or lysogenic pathway. Inside the cell, the circular lambda chromosome, when enter a lytic cycle, it reproduces and encodes enzymes that lyse the host cell, just like bacteriophage T4. But when it enter a lysogenic pathway, then lambda chromosome is inserted into the chromosome of the host bacterium and is replicated along with that chromosome. In this integrated state, the chromosome is called a prophage. The genes of the prophage that encode products involved in the lytic pathway, for example, enzymes involved in the replication of phage DNA, structural proteins required for phage morphogenesis, and the lysozyme that catalyze cell lysis must not be expressed because these enzymes and proteins are not required in lysogenic pathway. This figure shows life cycle of bacteriophage lambda. Firstly, lambda phage attach to E. coli cell and inject its DNA. The linear DNA of lambda phage converted into circular form inside the E. coli cell. Then this circular lambda chromosome can either enter the lytic pathway or lysogenic pathway. Lytic pathway is similar as discussed in bacteriophage T4, and it results in cell lysis and release of viral particles, while in lysogenic pathway, lambda chromosome is inserted into the E. coli chromosome, resulting in the formation of lambda prophage. Then, when the E. coli cell divides, then this lambda chromosome also gets replicated along with E. coli chromosome. Let's summarize what we have studied today. Bacteriophages are viruses that infect bacteria. Bacteriophage T4 is a lytic phage that infects E. coli, reproduces, and lyses the host cell. Bacteriophage lambda can enter a lytic pathway like T4, or it can enter a lysogenic pathway during which its chromosome is inserted into the chromosome of the bacterium. In its integrated state, the chromosome is called a prophage and its lytic genes are kept turned off. Thanks for watching the video. अगर आपको वीडियो पसंद आया हो तो प्लीज़ लाइक, शेयर एंड सब्सक्राइब.